Good afternoon, everybody. Good afternoon and welcome. Just delighted to see all of you who are here in Rackham and those who are joining us virtually for a very special occasion and event that I've been looking forward to. I'm Susan Collins. I'm Provost and Professor of Public Policy and Economics. And again, it really is a pleasure to be launching this special event this afternoon. We're gathered here, as you all know, for the Distinguished University Professorship lectures by three of our very distinguished colleagues who I'll have the opportunity to introduce to you this afternoon. I do want to say that occasions like this really have a special meaning in the Academy. We're taking the time together to reaffirm our commitment to the importance of ideas and to being a community that encourages deep inquiry and thoughtful discussion. And as I was saying to our distinguished lecturers just a moment ago, to me, those activities really bring joy and a reminder of what brought so many of us into the work that we do and inspires us to continue learning and working together to really move our ideas, our thoughts, our ways of having an impact forward. So this is essential work of the academic world. It shapes our research activities, our educational programs, and our service to the people of Michigan, the nation, and more broadly to the world. And so it's really good to have this public and celebratory acknowledgement of these values and of all of us who really infuse and carry them forward. So today, I'm proud to begin by saluting Donald Zack and Arthur LUPIA, as well as Nancy Love. And in doing so, we celebrate the very best of our academic community, and we honor the intelligence, the curiosity, and the commitment to learning that are the foundation of what we do and our mission. Today's lectures are being delivered in the Rackham Amphitheater, and it's one of many lovely spaces in a building that was designed to be a place for cross-disciplinary conversation and learning. And I have to say, it's one of my favorite places on campus, so delighted to be here this afternoon. In that spirit, I do want to note that our speakers come from different disciplines, and yet they share a commitment to multidisciplinary work that addresses complex societal concerns, so we're really fortunate to have this opportunity to learn from each of them. So first, it's my pleasure to introduce Donald Zack, Professor of Natural Resources in the School for Environment and Sustainability, and Professor of Ecology and Evolutionary Biology in LS&A. Professor Zack is one of the nation's foremost ecologists and a world leader in the study of soil microbial communities. His work is a major contribution to this field, and it also is his starting point for developing a critical new area of research that examines how organisms and ecosystems respond to global environmental changes. Among the concerns he explores are the impacts of enhanced nitrogen deposits, CO2, and ozone enhancement, and biodiversity loss. A thoughtful researcher who embraces new methodologies, Professor Zack develops and supports scientific curiosity among his students. He's a skilled teacher at all levels, with an impressive ability to combine lectures, lab work, and field experiences in ways that help his students to integrate theoretical concepts and also understand their real-world applications. Professor Zack has shouldered a number of leadership responsibilities, including the founding of a critical journal, extensive service on national review boards, and redesigning educational programs. And in each of these roles, he has helped to ensure that excellence will flourish in research and in education. In recognition of his outstanding scholarship, teaching, and service, the university is proud to name Donald R. Zack the Alexander H. Smith Distinguished University Professor of Ecology. And so it's my great pleasure to invite him to come forward and share some of his insights from changes that are occurring within the environment.
Thank you so much, Provost Collins. And this, too, is my favorite space on campus, and I'm so happy to be here talking to you this afternoon. I'd like to thank you all for being here and all of you for being here virtually that I can't see right now. Before I begin, I'd like to talk to you about Alexander H. Smith, because I think he was really a remarkable person and teacher. Alexander H. Smith was a professor here in, back then, the botany department. And his focus was on soil fungi. He's probably the most preeminent fungal biologist of the previous century. He identified and described more species of fungi than anybody in the history of the science. And he collected over 100,000 specimens of fungi that now reside in our herbarium, making it world-renowned as a repository for biodiversity and for DNA that can be used for biodiversity studies. He was a profound believer that teaching in the field, active learning, was the way to understand how organisms work in the environment. And that is something that I follow in my own teaching. His legacy lives on today in the Smith Foray, which is a gathering of fungal biologists in the Upper Lake States region every fall. A couple of years ago, it occurred at our biological station. Like Alex Smith, as he was known, I too am interested in fungi, but more broadly, microbial communities in soil. And this simple model has been the guiding post for the research that we've done in my lab over the past 30 years. And it's based on a very simple idea, and that is that plant and microbial communities are linked together in terrestrial ecosystems through the reciprocal exchange of growth-limiting resources. Plant growth broadly over the planet is limited by soil nitrogen availability, and nitrogen is made available by the microbial decay of dead leaves, roots, and soil organic matter. In turn, microbial communities in soil, their composition, who's there, their function, what they're doing, and their diversity, is influenced by organic matter inputs to soil, which are a source of energy for microbial communities in soil. Fungi, bacteria, actinobacteria. That source of energy, organic energy, in turn structures their communities. So plant and microbial communities are tied together in terrestrial ecosystems. And this simple ecological relationship allowed human civilization to flourish 10,000 years ago. This is the basis for agriculture, pre-industrial fertilizers, or mechanization. We've depended on it for millennia. Currently, we are modifying this relationship in an unconstrained global experiment. Atmospheric CO2 is a positive effector of plant growth, whereas ozone, ground-level ozone, damages leaves and decreases plant growth. We globally modify the diversity and composition of plant communities through all kinds of land use changes and manipulations. Globally, we have doubled the amount of nitrogen entering ecosystems from the atmosphere due to the burning of fossil fuels. It's a growth-limiting resource for plants globally. And we're altering temperature and precipitation regimes. We are twisting all the control knobs on these communities in a global way that we really don't quite yet understand. And we have sought to understand this basic ecological interaction as well as how it's being influenced by human activity. So the question is, how will microbial communities in soil respond to these global environmental change aspects? And there's many of them that we don't understand, and it's been difficult to pull them apart. Now, I want to first talk about microorganisms on this planet, and then I want to focus in on some particular aspects of the work that's been conducted in my lab. Here's the Tree of Life. It's a cartoon. I pulled this off the web from the Tree of Life website. And there's a lichen, there's a rose, there's a caterpillar, a mushroom, and it's a cartoon because this is the real Tree of Life. This is the molecular Tree of Life constructed from ribosomes. They're subcellular organelles that all organisms on this planet have. And we can understand the evolutionary relationships among organisms by using this approach. Now, I want you to notice these two withering sprigs here. That's all animals and all plants on the planet. Everything that we have learned virtually about the ecology of organisms has come from those two sprigs. The length of branching and the amount of branching is an indication of diversity. So you can see that the vast majority of diversity on this planet is not in the plants and animals around us, but in the things we can't see. Now, this is audience participation time. These two things look the same, but they're really different. What are they? Tell me. Come on. Call it out. Stars in the universe. Well, it's the other one. What's the other one might be? Nancy? It is. They're fluorescently labeled microbial cells. Two vastly different scales. So, good job. Microbial cells and stars. So, what's more? Stars in the universe or microbial cells on this planet? Raise your hand for stars in the universe. This is a leading question, right? Raise your hand for microbial cells on Earth. Okay, very good. So, there's 4 to 6 times 10 to the 30th cells on Earth. There's 1 times 10 to the 28 stars in the universe, 2 orders of magnitude greater microbial cells on Earth. Now, here's a quote that's been attributed to Da Vinci. We know more about the movement of celestial bodies than about the soil underfoot. I'm not sure if that is accurately attributed to him, but it's attributed to him in the literature. And I will argue that this is true today, despite the fact that we are so dependent upon this fundamental ecological relationship for food and fiber throughout our planet. So, what have we learned about microbial life in soil? We've learned that there's a high level of diversity in soil, and that a gram of soil, about a pile the size of 1/4 in your hand, contains about 1,000 to 10,000 species of bacteria, about 100 to 1,000 species of fungi, and about 10,000 species of archaea. These occur across the entire domains of life. Moreover, what we know prior to the advent of molecular techniques is only 1% of these organisms. So molecular biology has revolutionized our understanding, and within my professional career, we've gone from 1% to really understanding a great deal about the diversity of communities of soil. So all of you may think that this is the most diverse type of ecosystem on Earth. You now know that's wrong. It's the soil underneath your foot that is the most diverse habitat on Earth. And yet we have so much to learn about the ecological interactions that generate the composition, who's there, the function, what they're doing, and the diversity of these organisms. So I'd like to talk to you about 2 large-scale long-term experiments that I've been a part of to understand how our activities are modifying this basic relationship. And the approach that we have taken is that we can understand a biogeochemical process, like the cycling of carbon or nitrogen within terrestrial ecosystems, by understanding the enzymes that mediate that process, and then today being able to look at the genes that encode those enzymes that do those biochemical tasks. We can obtain that genetic information out of soil and link who's there to their function and to ecosystem level processes that are of global significance. So I'd like to show you one experiment that we conducted with a team of scientists and graduate students and postdocs to understand how the rising atmospheric CO2 and ground level ozone concentrations are going to alter this feedback between plant and microbial communities in terrestrial ecosystems. Now, there's been an idea in the literature that because atmospheric CO2 is a fertilizer for plants, that they will grow more, deposit more leaves and roots on soil, that will slow the decay of organic matter and tie up nitrogen in soil organic matter, making it unavailable to plants, thereby downregulating the ability of plants to grow faster at higher CO2 concentrations. That would be an issue because it means as we continue to increase CO2 in the atmosphere, terrestrial ecosystems will dampen in their ability to store that carbon. We conducted this experiment in northern Wisconsin, and these kind of look like high-tech Stonehenge rings. And what they really are free atmospheric release experiment rings that waft a drifting cloud of either ambient air, elevated CO2, elevated ozone, or both through developing forest that's planted within them. So the vertical stanchion pipes have valves going up them that release these gases. There's a weather station in the center that looks at wind speed and direction, talks to a computer, and wafts CO2, ozone, both, or ambient air through this developing plant community, which we started from bare ground. We added a tracer, nitrogen 15. The majority of nitrogen in the universe has a mass of 14. There's some nitrogen that has a mass of 15. We can use that to follow the flow of nitrogen in that reciprocal relationship that I talked to you about. We added this tracer to this experiment, and remember the prevailing idea had been that greater plant growth at elevated CO2 would deposit more plant litter, would slow decay, and that would grind the nitrogen cycle to a halt, keeping plants from responding to atmospheric CO2. So I just want to remind you of that. And here's what we found. So this is the amount of nitrogen in all of the plant tissue that grew after 12 years. We harvested all the above ground portions of these trees and their below ground and analyzed them. There's more under elevated CO2. More than that, the trees took up more of this isotopic tracer. These observations don't support the idea that plant growth is going to grind to a halt as CO2 accumulates in the Earth's atmosphere. It suggests that forests are going to develop at a faster pace as this trace gas accumulates. The deposition of nitrogen from the atmosphere is another global change. Organisms at this point in time are seeing more nitrogen in the environment than they have during the entire course of their evolutionary history. We've doubled the amount of nitrogen falling out of the atmosphere since pre-industrial times. And again, it has the potential to alter this reciprocal relationship between plant and microbial communities. From first principles of physiology, we know that there's a group of fungi in soil that do the hard work of decaying woody material. And under lab conditions, if nitrogen availability is high in the environment, they turn off the transcriptions of the genes that encode enzymes to do that hard work of breaking down woody material. We reason that this could be an important phenomenon because global models suggested that there's an unaccounted for sink for carbon in terrestrial ecosystems. We can't balance the global carbon budget. And moreover, that sink occurs in northern temperate forests where rates of atmospheric nitrogen deposition are very high. So we set out to test that idea. And we did that in a series of 4 forest stands that span the lower and upper peninsula. They're all dominated by sugar maple. They're the same age, and they occur on the same soils. and we added additional nitrogen to them in amounts that are likely to occur by the end of this century. So an ecologically realistic amount. And we did this for 22 years with support from the National Science Foundation and Department of Energy. And here's what we learned. That peroxidase, the enzyme that does that hard work of breaking down woody material, its transcription, gene transcription, is downregulated in these soils. And moreover, at the same time, soil organic matter storage increased, suggesting that this could be an unknown sink for atmospheric CO2 in terrestrial ecosystems that's of global importance. So, I've shared with you the conceptual model that underlies all of our work. And I've shared with you the approach that we've taken by which we can link the regulation of fungal metabolism, bacterial metabolism, to biochemical processes, to the cycling of carbon and nitrogen at a global scale. It scales from genes to the globe. Now, this work would not have been made possible without the numerous undergraduate students, master students, doctoral students, postdocs, and research staff that I've been so fortunate to work with over the years, in addition to the marvelous colleagues I've had here at Michigan. So thank you again, and I appreciate you being here today to hear my talk.
Thank you, Professor Zack, for that wonderful lecture. We have time now for questions from the audience, both here in the room and also virtually. We have a way to receive questions from both, so we have that available, and we have microphones that we'll bring around to you. Please use the microphones for your questions, since that will make sure that our virtual audience gets them.
Thanks, Don. That was great. When you talked about the peroxidase downregulation and the increase then in the resilience, I guess, of that material, I was thinking negatively about the fact that our waste build up and we're not putting that back into the soil then. You're thinking of it as a carbon sink as well, which is maybe a positive, but isn't there a negative consequence to that? Can you comment on that?
Oh, yeah. Sure, Nancy. Nitrogen deposition is not a good thing. It may, in the short term, increase ecosystem carbon storage, but that excess nitrogen is leaking into groundwater, into surface water. It's part of the issue that's in the western basin of Lake Erie. Not a good thing, and it's not sort of in a Gaian way, some kind of self-homeostasis regulating mechanism. You know, I lecture every day with the mask on, and I forgot I could take this off.
There's it. There's a question?
Yeah, thank you. I think this relates to the previous question, and that was back to the CO2. And you mentioned about, if I understood it correctly, increased CO2 will lead to an increase in the growth of plant life. I imagine there's not a free lunch there, right? And there's going to be some tipping point. What is the downside to that?
Oh, the downside to more CO2 in the atmosphere is that the planet continues to warm. And you know, the real question is, in my mind, do trees grow faster or do they grow bigger as CO2 accumulates? And the distinguishing between the two is really important. If they grow faster and attain the same size, then terrestrial ecosystems are not going to be an additional sink for what we're putting in the atmosphere. If they grow bigger, they will. The problem is that, as you can see, these trees were 12 years old when we harvested them. We can't do that experiment. It's happening in real time.
I have a question I would like to ask, which is this side comment you had early on that only 1% of microbial species are culturable, and it seems like you're actually doing genetic analysis then in your later experiments. How has that gap, if you like, affected the work that you can do or want to do, or how you've gone about addressing that big issue?
It enables us to do work that we couldn't any otherwise be able to do. And it's more affected my teaching than anything else. I can tell you that my soil microbial ecology texts that are on my shelf from when I was in graduate school are just flat wrong about some things that we've learned. They'll tell you that Streptomyces is the most important actinobacteria in soil. But when you look at DNA that you get out of soil, it's a small fraction. It just turns out that it's the one organism that most readily grows on laboratory media. So in terms of research, it's been enabling. In terms of teaching, it's been a point of revision.
We have a question there.
Really fascinating work, Dawn, and I'm wondering if you've looked at all at the biodiversity of the soil microbial community in these two experiments where you alter CO2 and ozone and nitrogen.
We have, using the techniques that we have, we are able to look at a number of aspects of diversity. Species diversity, functional diversity, which genes are present that do which jobs. And so this has really opened our eyes into what's happening in soil. Soils are, as I showed you, are hyper-diverse. And one question that we just don't have an answer to from an ecological perspective is what fosters that diversity? And it's something that I think about almost every day. I mean, we don't have the theories to be able to generate that much biotic diversity in soil. So understanding it remains, I think, a really exciting and important contemporary challenge.
Thanks, Don, for a lovely talk. Most of these questions have been about the DNA revolution, and I think that's probably been the thing that's changed the most over your career. But I wondered what's second? Like, what is the biggest change in your field, aside from the DNA revolution, allowing you to explore this microbial biodiversity?
I'd have to say that is really the overriding factor that's revolutionized the work that we do. When I was a graduate student, it was a pipe dream to think you could get DNA out of soil, let alone amplifying particular genes that do particular biochemical tasks. I guess the other thing that would come along with that is this work generates a mind-blowing amount of data. And the ability, the bioinformatic ability to deal with this plethora of data and diversity has also revolutionized it. They go hand in hand.
I think we have time for one more question. Okay, I'm going to try to sneak it in then. So very impressed with the long time scales of the experiments, 12 and 22 years. I'm thinking of some of your graduate students, yourself. Can you just say a little bit about how you've kind of sustained like the vision of the work, or how what is happening and why you're waiting those 12 years to get those extraordinary plots? Could you talk a little bit about that?
That's a great question, Mike. And being here at Michigan, I'm privileged to have an endless supply of really outstanding students, some of which are in the audience here. And so that has really sustained this effort. And what's financially sustained it is the good fortune to have repeated funding from the National Science Foundation, from the Department of Energy to sustain these experiments. And so all of this work is done on 3-year increments from both of those agencies and putting it together into this long-term record.
Well, thank you very much for a wonderful lecture. Please join me in thanking Distinguished University Professor Donald Zack for his lecture.
Let me add my thanks and congratulations as well. Professor Zack, Don, that was wonderful. It is now, and I will take it off. It is now my pleasure to introduce Arthur LUPIA, Professor of Political Science in LS&A and known to many of us as Skip. He is internationally recognized as an innovative thinker whose work breaks new ground in understanding elections and political communication in the United States as well as in other countries. One of the key questions that he explores is how citizens make voting decisions in democratic societies. His research illuminates the processes through which voters take cues from others, particularly knowledgeable elites, as they choose among candidates or policy options. This work deepens our understanding of areas such as public opinion, direct democracy, digital media, legislative organization, and policy implementation. Professor LUPIA's embrace of varied methodologies as he explores complex questions has added significantly to the study of politics in the 21st century. An enthusiastic teacher, he excels in large introductory courses and as a mentor of graduate students. His commitment to teaching and preparing students for citizenship led to the creation of a new course, Beyond Partisanship, that helps students recognize and move beyond partisan positions, their own as well as those of others. Professor LUPIA is a dedicated and thoughtful advisor to the National Science Foundation, providing leadership for its social science division. He's also led the American National Election Studies, and under his direction, it became more inclusive of Black and Latino voters and developed accessible and engaging communication platforms. The university is pleased to recognize Professor LUPIA's contributions to our understanding of the political world by naming him the Gerald R. Ford Distinguished University Professor of Politics. And now I'm delighted to welcome him to the podium.
Thank you so much. All right, good afternoon. Oh, I can take this off, right? Good afternoon. What I want to talk about for the next couple of minutes is you. And everyone in this room and at universities like this. Every day we have an opportunity to improve the quality of life of others. Not just the people we can see, but people who will never set foot on a college campus or never see the Block M. And there's urgency in the things that we can do for people. But there are also challenges. As you may have heard, we live in an era of misinformation, where alongside our ability to communicate long distances through video, there's a counter-narrative where people present different types of information. So the question is, what have we been doing well, and how can we do better? I think we all believe that we can do better, but how do we do it? So let's run through that. An appropriate place to start is with gratitude. So I do want to acknowledge the leadership of this university. Many of you are in the room here. It's an incredible, I was in Washington for a couple of years. Watching the leadership of this university from that vantage point was really, it's an amazing thing and not to be taken for granted. The faculty here, the amazing things that are being done, the students and the alumni worldwide. This is an unbelievable institution whose reach is felt, whose impact is felt on a daily basis by millions of people around the world. I also want to acknowledge President Ford. President Ford was an incredible public servant, dedicated throughout his life, humble, known across both parties for his honesty, for his integrity. It's an unbelievable thing, and I think he represents so much of what we want to see in all of the graduates of the University of Michigan. I'm very proud to have this association. But I want to focus on science and what science is and what it can be. I like to think of science as service, a way of knowing where we can help people reconcile their aspirations and the actions they can take to help them figure out how can we do better tomorrow than we did today. I also think about science as a basis of innovation. And by innovation, it includes starting the next tech company, but it also includes the community that's next door to ours or next door to any of ours, where we can help people think about how do we build the tomorrow we want from the today that we have. Right? So it's an incredible thing, the shared endeavor that we have to try and produce rigorous and precise research that empowers people and improves quality of life. But there are challenges, and they're pretty big ones. We are in the middle of a pandemic, and the numbers can be overwhelming of the people who are affected, of the people who've died. Every one of those persons had a family. Every one of those persons was part of a community, right? Anything we can do to help communities, to help humanity through these things, it's important that we do so, and that we do so as well as possible. But we're not just in the middle of a pandemic. An economic transformation is also occurring. It was happening before the pandemic. But so many people, so many communities, the jobs they had, the industries they believed, the life plans that they had have been disrupted by changes to the economy. There are great new opportunities, but it's not clear that they're available to everyone. On top of the pandemic and on top of the economic disruption, there's disruptions to every education at every level. And I think everyone in this room has experienced that. But every day in education means so much. Every moment that we have with a student, right, is an opportunity to change their world, to broaden their horizons. And finding new ways to do that in our changing circumstances is very important. And then on top of all of this, in this country and around the world, injustices have come to light. Many of them existed for years, decades, centuries. But now we are realizing just how pernicious and pervasive they have been. And we're having a moment of reckoning of how do we do this. So these are all challenges that are in front of us. And they're significant. Today we are challenged in terms of our bodies, in terms of what a pathogen can do to our bodies, right? In terms of our minds, in terms of what changes in the economy and changes in education can do to our opportunities. And at the same time, we're challenged in truth. On so many issues now, there are different points of view. If you log on to social media or if you look at cable news, there is no shortage of experts who will make claims about where we are and why. And so truth itself is challenged, and the question is, how do we respond? So what's fascinating is that even in the darkest times, if you looked in the right places, you could see people trying to build a better tomorrow, right? So when we were faced with questions about, well, why should we pay for expertise? There were people answering that question through their work. When people were asking questions about, yes, but why should I trust you? You're an elite, you are different from me. Why should I base what I'm doing on what you're doing? If you looked in the right places, you found people, even in this environment, doing incredible things. All over the world, from the early days of the pandemic, people were trying to figure out, how do we manage this new reality of relationships, where we've built our whole lives around the idea of being close to one another, and now we can't? How do we think about new economies? How do we think about translating in-person education into hybrid? But if you look closely, you saw something else too. You saw people in certain places trying to create light out of darkness, right? You saw people looking at confusion and creating clarity, right? You saw institutions all over the world that are trying to make a difference. And this institution, the finest public university in the country, you had people in every field, right, drawing from previous innovations and racing to create the next innovations that help us this week, this month, and beyond. And we can't lose sight of how important that has been, but the challenges remain. And so what I want to talk about in the last couple minutes is, how can we do better? How can we achieve more of the potential that we think science has, that we think universities have, in an era of misinformation? So I want to propose 3 adaptations, 3 things that I think we ought to think about seriously to increase the public value of a wide range of efforts. And one is greater transparency. This matters for science, and I'll walk through why. A second is better communication. We could talk for a long time about this. I'm going to give you a 3-minute overview, basically of how not to get in your own way when you're trying to communicate a complicated topic. And finally, more effective engagement. If what we're really concerned about is the public value of our effort, it can't be a one-way street, and everybody knows that. But the idea is, how can we engage more effectively with the people that we're trying to serve? So that's the roadmap of where we're going next. Greater transparency. So what's fascinating about science is one way of knowing. There are actually 4 ways to validate a knowledge claim. If I say that Tom Brady retired, I'm saying that Tom Brady is in the set of retired. And there are 4 ways to validate that claim. One is I can validate it through metaphysics. I can say that there are forces beyond our ability to comprehend, and they make certain things true or false, right or wrong. And some people among us have special access to that, and we can learn from them. And that manifests through religion and faith communities and theology, and throughout human history, it's been a powerful way of knowing. We can also say, well, no, we can learn from culture. We can learn from art or literature or architecture. There are things that come from the past that are encompassed in these forms, and we can look at them, we can read them, we can experience them, and maybe they can guide us about what to do next. And then, of course, there's personal experience, the idea that someone we know experienced something, and they tell us what it looked like to them and how they felt about it. And we can learn from that, too. And these are all powerful ways of knowing. And then there's science. And what's amazing about science is a commitment to a set of practices. Science is a commitment to a method. And if we do it the right way, the validity of the claims we find hold regardless of variations in your theological commitments, regardless of your cultural commitments, and regardless of the diverse personal experiences that you've had. Science isn't the only way of knowing, and for many of our biggest questions, we need them all. But science gives us this opportunity to find validity that might hold for all of us. And so this matters today because when most of our scientific institutions were formed, people couldn't have science. You couldn't get it. In 1950, when the National Science Foundation was originated, it cost roughly $7 to make a 10-minute call from New York to Los Angeles. In 2022 dollars, that's $77 for a single 10-minute phone call. So for those of you complaining about the price of your unlimited plans now, let's put that in perspective. But this is reflective of the idea that if you wanted information or expertise on almost any topic for almost anywhere in the world at almost any point in human history, you couldn't get it. The average household in 1950 had about 12 books. If you lived in a city, you can go borrow 1 or 2. If you lived in the country like I did, good luck, right? So expertise for most of human history was, thank you, where else am I going to get this information? Right? Airfare, the same thing, 1955, right? You want to fly from Chicago to Phoenix, $138. In current dollars, that's about $1,400. People didn't travel. People didn't go across town to get expertise. All of the academic institutions, scientific institutions we know about, were created in an era where expertise was really scarce. If we were asking why are there all these questions now about whether we should pay for us or whether we should trust us, part of it is we've got so many people who are experts now. We can get information for free everywhere. That's our reality. So the question is, how do we respond? And in science, I think there's one response. And that is, if we're going to say that science shows something, we really have to commit to the method. There are incentives to exaggerate. There are incentives to overclaim. And every time we do that, we feed a narrative about science being something different than it actually is. Science can be a source of intersubjectivity, of intercultural validity, of validity that applies across faiths and circumstances. It's a powerful thing to have in addition to knowing. In addition to other ways of knowing, it's critical that we're as transparent as possible about what we've done. Communication also matters, and I'm going to give you a shortcut. We have a theory about how people learn about things like science, and the idea is there's this thing called truth, and we convey it with great force and vigor, and it flies through the air, and it goes into the brain of another person, and they are educated. And so if you've been paying attention for the last 15 seconds, what I want to tell you is every single sentence I said was false. That is not the universe into which we were dropped. Learning doesn't happen that way at all. But there are some things about learning that can really empower us to convey complicated things. And so I'm going to give you one piece of bad news, and it's going to be followed by good news. So the piece of bad news is I can't teach you anything. All I can do is make variations in sound and light. I'm making variations in sound with my voice, and I'm making variations in light with my hand, and by putting things on the screen. To the extent that learning occurs, it's because an audience member is so captivated by the variations in sound and light that they work to take those variations and create memories from them, work them into a conceptual framework that they have. That's how learning works. Unfortunately, it's the only way that it works. So I'll give you a more concrete example. If you think for a moment, if you have a favorite song and you think about it, if you can close your eyes, and if you can hear it, I know a couple things about what you're hearing. I don't know exactly what song it is, although if it is in the pop music vernacular, I know probably most of the lyrics are wrong. Watch YouTube, you'll find this. But the other thing that I know is if you can hear a song, probably the time signatures are different. Some of the instruments are missing, right? Some of the melodies aren't there. But you can create it, and it's your own. Because the version you can hear, you have co-produced. When you have a memory of a movie or a picture, you have co-produced your own version. Learning requires co-production. There is no other route. So as we're trying to teach people about science, the question is, how do we create the conditions for effective co-production? And to make a long story short, the idea is communicating with an audience rather than to them. Understanding that the most interesting parts of a learning act happen between the ears and slightly above of the other person. It's creating a circumstance where what you're providing to them is the next step on their journey. The ideas that hit their critical concerns and help them figure out how they can build a better tomorrow from today. We can talk a lot longer about this, but the other thing I'll tell you about learning is whatever you think human attentive capacity is, divide that by a large number and you're closer. So this is our route, and we have the ability to every day to do a little bit better. So finally, I want to talk about engagement. Ultimately, the value of what we do, a lot of it's realized by others. It's amazing to discover and collaborate with everyone here. That's great. But when we think about our public obligations, the idea is, well, what matters for them? And this is a hard question to answer. I just came from 3.5 years at NSF, and this is something we dealt with every day. And what I want to show you is how we dealt with it. Because at NSF, we don't fund applied research. We fund basic research. Ask me what that means later. But it's hard to explain, well, why is it urgent to fund this? And so we came up with a framework, a way that anybody at any stage in a research process could try and answer the question, what is the public value of this? So it's called the Broader Impacts Framework, and if you type NSF 21059 into Google, you will find it. But it's 3 questions you can ask yourself about what you're doing. And the first question is, your work, when you do science, it creates scientific opportunities, like grad students get to do things, you get to do things, and communicative products, articles, videos, syllabi, books. Who do they empower? Who can do something tomorrow as a result of those that they couldn't do today? If you're thinking about that from the very first stage of a research process, it can help orient us doing work that not only advances science, but has a greater likelihood of creating value. Second, if people are empowered, whose quality of life does that empowerment improve? And again, this is something at NSF I was forced to think about all the time, because we would be going to Congress on a regular basis, and they'd say, why should we pay for science? Why should we pay for this field or that field? And it's important to make the connection, even for abstract research, to talk about the possibility of what discovery today could do for people tomorrow. And finally, what actions make the quality of life improvements more likely? I think anyone can do this. And if we do it a little bit, it helps us answer questions. It helps us inspire people to do work that at the end of the day does all the amazing things we want science to do, but also empowers others. So through greater transparency, we have a commitment to honesty. Through better communication, we have a commitment to delivering ideas that give back. And through thinking about engagement, through thinking about the people on the other side of our scientific workflows, we have an ability to reconceive how to create value to them. And so I think that's where we are today, as we think about the public value of scientific research in an age of misinformation. Misinformation isn't going away. But we can do better at creating value from what we do so that more people, when they look at what we're creating and what else is out there, they can make a decision that's best for them. And that decision is ours, right? So every single day, for any of us, creates this opportunity to conduct research that empowers people and improves quality of life. And so when it comes to how can we battle misinformation, there are some clues here, but to make it more simply, we should just seize it. We should seize that day. And there's no better place in the country to do that than at this university. So thank you for your time and your patience. Go Blue.
Thank you for that lecture. We have time for questions. Yes, we have individual there. And just a reminder, you can also, if you're joining us virtually, you can ask a question into the chat and it will make its way to the speaker. Please go ahead.
Yes, thank you very much for your presentation. I think during the pandemic, I've never heard the word science used as much as I have on a daily basis. But yet, most people really don't know what the word science means. So the default position seems to be you sort of accept it, you cannot question it, yet that's sort of the antithesis of the scientific method. So how can educators going forward educate the public as far as what the term science actually entails?
So it's a fantastic question, and let me unpack it just a little more, right? Because in many societies, science rose, and sometimes it was a challenge to like theological ways of knowing. But now we have the irony, since many people, won't look at the lab book, won't read the proofs. At some level, we're asking people to take science on faith. So there's an irony. And the question is, why might people want to take us on faith? Because we're all human beings, and we're amazing, and we're my favorite species, but we're also highly imperfect. And what the scientific method does, right, is it gives us a guide to identifying relationships and a way to explain them such that we can convey the validity to others very clearly. So in terms of teaching science, I think the most important thing to teach about science, just asking me, is about those values, is about adherence to the scientific method, however it manifests in your field. What is harder to do is come up with like a global science exam that will please everyone, because biology is like critically important, mathematics critically important, engineering critically important, linguistics critically important. I'm not sure what the intersection of essential knowledge for those four fields are, so I don't know that I could come up with the test. But if you give me the opportunity to say, to the extent that you care about promulgating a form of knowledge whose validity is almost entirely independent of cultural variations, personal circumstances, or theological variations, right? Adherence to the scientific method is critical. Not exaggerating is to get attention, to get in the media is critical. Because the cultural authority of science, which even with all the questions we still have today, it's an amazing asset for this country. The idea that there are so many people who at the end of the day, they won't read the proofs, but they really want answers from us. I mean, think about where we would be without that. So, I think teaching people about, the other thing I'd say, and it's easier to do at a public university, people pay for this. People pay for this building, people pay for our research, and they're real. And they have real challenges, and they have real problems. And we know at this university that part of our job is to serve them. I think, reminding students that, when you're doing science and it's funded, the discovery part's cool, but the service part, it's a critical part of what we do, what sustains us.
Thank you very much for that lecture. Taking your paradigm and applying it to an issue that's front and present right now, vaccination rates. How could we have done a better job, or how could the government have done a better job, achieve a higher vaccination rate in terms of explaining the process, communicating the science, or engaging the public? When you look at vaccination rates in many other countries, they're much, much higher than ours. So what could we have done differently? Yeah,
that's a really good question. What I will tell you is I had a front, I was at NSF when everything happened, and I'm not answering your question, I'm answering a slightly different question. There's the government that you see on TV, and then there's this amazing thousands of people inside the government dedicating themselves to how to make life better, how to do better today. And the way that even under different presidential administrations, the mobilization of expertise that happened very quickly was astonishing. All right, so I will say that. But in terms of how to do better, with information, particularly on complex or scary problems, we might like to think that we process information by thinking about it first and feeling it later. But in fact, human information processing is feeling it first and then once in a while thinking about it. But mostly it's feeling. And so what it means in cases like vaccination is if I walk into a room and I threaten you, I can do it by verbally threatening, or I can just do it by kind of giving off an air of superiority or signaling that I don't really share your values. Combining that with limits in human attention, the next 10 minutes of things I say could be entirely factual. But if I've presented the information in a way that's threatening to a potential audience member, they can't hear me. In fact, I become kind of like Charlie Brown's mother, right? And if you're not sure what that experience is like, I don't know what your ideology is, but think about what your ideology is for a moment, and I'd ask you to watch the cable news network tonight that is of the opposite ideology. And the question is, when you turn that on, is it like, okay, I'm really going to give this, I'm going to think about everything they're saying and really give it a chance? Most people are like, keep me away from this. So when we approach people, the key thing we have to do is find common values. That's the key way to approach, to understand why someone's concerned or what they're trying to do in the day. And what you can do is walk people through things we can agree on, And where that gets us with conversations about vaccination is some people will move. Some people you can't move. We can't get to 100%. But if you're asking what we could have done differently, and there's great research on this, because vaccination has been an issue for a while. I mean, it's more so now. If I walk into a room and talk about anti-vax people, I'm a dead letter. Like, I will, the people who already believe it, yay team. And the people who are not sure, I've just made myself the enemy. The same thing is true on issues like climate change. Right? There's been great work on climate change, particularly with people who, there's an association between skepticism on climate change and being an evangelical. Right? But there's been great research and practice on how different approaches to this problem has led to a huge movement within the evangelical faith to say, you know what, part of our faith is being stewards of God's creation, and that includes the environment. And so now you've taken this resistance and turned it into, I'm interested in science too. So that's a little bit of a framework. Thank you.
I have a question that I'll ask. Kind of really interested in this kind of role of co-production that you talked about in learning, and was thinking about that in the context of transparency, that there's a way in which the public could co-produce the knowledge if things are sufficiently transparent. But I was struck by how many steps there were in that path to transparency on that slide that went by. If there were one thing that an institution like ours or researchers in this institution or learners could be thinking about in terms of transparency with respect to the public, like what would be that thing to do? Or do we need to do all 8 of those?
So as tempted as I am to have a one-sentence answer, in different fields of study it manifests, right? So in a field like social psychology, replication is a big deal. I mean, there's actually been news about that, right? And so much of that is because we see a study in social psychology and we want to apply it to improve education or counseling or something like that. And so replication really matters. But if we're looking at astrophysics, it's really hard to replicate LIGO, like the observatory. And so there's not sort of the same advice, but when people ask me, I want people to be as truthful as possible about the process that has led to their discovery. And some people will only want the 5-second version of, well, what's the answer, right? But other people who want to look more deeply, if I give them access to it, they can figure out whether my finding applies to them or not. And those things happening simultaneously then builds this broader set of people who make informed decisions about whether or not to trust me. And at the end of the day, coming back to the question about the cultural authority of science, the more opportunities we have to demonstrate that trusting people who follow this process can lead you to actions that help you achieve your dreams and aspirations, right? That's ultimately, I think, how you build the support.
There's no further, let's thank our distinguished university professor for the lecture. Thank you.
Thank you.
My thanks as well to another really excellent lecture. Thank you very much. And last but certainly not least, it is now my pleasure to introduce Nancy Love, Professor of Civil and Environmental Engineering in the College of Engineering. Professor Love is an environmental engineer working at the interface of water, infrastructure, and public health. Her research group uses chemical, biological, and computational approaches to improve water systems. With her team, Professor Love leads research that evaluates contaminants in water and advances technologies to recognize and remove them, and seeks ways to recover resources from water. Her research is informed by and shared with water treatment professionals worldwide. This critical work is shaped by a concern for community understanding of and engagement with policies about water systems. Actively involving students at all levels of her research, Professor Love focuses on addressing public concerns while mentoring the next generation of experts on the complexities of water quality and management. Her teams are deeply engaged with the communities they serve from Flint to Addis Ababa. Professor Love is a skilled and innovative teacher, utilizing approaches such as peer learning that help students develop their skills in working collaboratively and learning from one another. She is also a dedicated mentor, committed to creating a diverse and inclusive engineering community. In addition to her leadership in research and teaching, Dr. Love provides exemplary service to her profession and to the university. She serves on national and international boards focused on water and the environment, and she has served in critical roles in her department and in the college. And so the university is so proud to recognize Nancy Love's contributions to environmental engineering, teaching, and service by naming her the Joanne Silverstein Distinguished University Professor of Environmental Engineering. And now it's my pleasure to welcome her to the podium.
Well, it's wonderful to be here. Thank you so much, Provost Collins, for that. I'm lucky because Joanne Silverstein is here. She's in the room. You can wave your hand, Joanne. And for those who are in Zoom land, you can trust me on this. I'm really tremendously honored to be able to name this distinguished university professorship after Joanne. She is the first woman in my field, in the water quality engineering field in academia. She's a decade ahead of me, and environmental engineering is a fairly young discipline. And so she is a person who really paved the way for women in our field and in engineering in general. She is an engaged person, intensely curious, committed to justice, and honors all of those elements in the way in which she has pursued her career. So she's a change agent. And I really wanted to honor her and her work through this professorship. She was the first woman department chair in the College of Engineering at the University of Colorado. I was the first woman department chair in the College of Engineering at the University of Michigan, acknowledging that Martha Pollack was an associate chair, functioning as a chair, and then became our provost and now is president at Cornell. So thank you, Joanne, for being here. Really, I'm glad you're able to share this moment. So I'm here to talk about the work that my students and collaborators and I have done with these water systems that the provost introduced me with. And I want to talk about kind of this process that I've gone through in my career. And I titled this Moving Outside the Fence, and it will be apparent in a few moments why I said that, but really trying to achieve sustainable and equitable access to high quality services, of water services. And I'm going to take you through 3 vignettes out of many I could have selected as I do this. I also want to acknowledge the funding for this work comes from a range of places, in both the NSF, but also industrial foundations such as the Water Research Foundation, and also some utilities as well, utility funders. So centralized water systems have a very substantial role in our existence as a high-income country. Centralized water systems have provided an opportunity for us to live in cities, close to each other, and in dense populations. Without them, we couldn't do that. And I would argue that economic prosperity that high-income countries benefit from can be achieved because we have these centralized water systems. So they have served us well. I'm going to talk about some of the challenges with them, but they have served us well. One of the things I'm showing you is this schematic here, is that these systems are complex. I'm showing you, we pull water out of the environment. We put it through multiple different reactors. And the reason we do that is because we have multiple different contaminants we're trying to contend with, and there's no one reactor system that does it all. Plus, we need redundancy, and we need to have protection in the event that one of those fails. And so this is called a multiple barrier approach, and it is the gold standard for the high quality water services we provide. We take that water, we pump it out, we distribute it through pipes that run underneath our streets, we use it in our homes, we discharge it through our bodies, through processing food, through doing laundry, showering, whatever, and it goes into a sewer, and then it goes into another part of the water system, historically called a wastewater system, now called a water resource recovery facility. Again, multiple barriers, multiple steps. And so these are complex systems, and my time early on in my career, first in practice, after my master's degree, I worked in consulting for a few years and did design mostly of the drinking water side of these systems. And so my fence is this box around the engineered system. And then when I moved into academia after finishing my PhD, I've spent most of my time in a very small part of this very complex system. Either in the drinking water distribution system or in this little bioreactor right here. And so much of my career and getting tenure was associated, and Don set me up for this, with the central dogma of biology. I too benefited from moving from culture-based methods to molecular methods, and was at the kind of early end of that in the 90s, where we were people were pulling DNA out of complex biological systems. Don talked about soil, a very complex system. So are the bioreactors, although not as diverse, I suppose, as his soil. But most people weren't doing that. They would do very simple, reductionist, simple science. I was trying to work in very complex systems and show we could get these methods to work. And I actually skipped the DNA part and went straight to proteins and metabolites first, before we had kits and all the really cool stuff we could buy now to make this easy. It was hard. And then later moved back and looked at DNA. So did a lot of stuff, learned about microbial functions, and learned about microbial structures in these bioreactors so that we could operate them better and use them to be sure that we removed the contaminants that we were trying to contend with. But after a while, I guess I started to realize that I was really focused on this very myopically on this engineered system. And it's a complex system, but the real complex system is much bigger. We have the water system and the wastewater system, and then we have the community. And the community has institutions, it has people, it has neighborhoods, it has ideas, it has regulations, it has all this stuff. And we can't think about our little engineered system independent of that. And so I've really spent time trying to understand the institutional complexities, the human complexities, the economic complexities of water systems on top of the technical complexities. And so I'm going to take you through 3 vignettes. And in each one of these, at the bottom, I'm showing the icon for the non-academic partner who's at the center of each of these. And I emphasize that because I feel like the timing is great for me to be the third after these 2, because I'm going to overlap with both of the former speakers in terms of the importance of how we communicate outside of the academy. So I'll start with the first one here, which is about the food nutrient cycle. And to start with that, these are graphics just showing you how nitrogen and phosphorus move through our human systems. We apply fertilizers, nitrogen or phosphorus. Oops, sorry, let me go back. How do I do that? There we go. You know, let's say 100 units. And then it's applied by the farmers, and we have tremendous losses through loss of crop, surface flows down into the soil and into the groundwater, tremendous losses of that nitrogen and phosphorus into the environment. Then a fraction of it ends up in the foods that come to market that we buy. They enter into our communities. We buy some fraction of that food because some of it's lost, and then it's consumed by us, and then it's sent to that wastewater treatment plant or the water resource recovery facility. And about 75% of the nitrogen-ish, you know, between 50 and 60% of the phosphorus ends up back in the environment in many communities in a reactive form. This is called a linear process. This is inefficient, and this is not sustainable for the long term. So what we think of this as kind of the linear movement of nutrients through our communities, what I was just showing you. And you can see that there's a tremendous amount of energy invested in creating those fertilizers, as well as the environmental impacts that I'm not even noting here. There's a lot of energy associated with that water resource recovery facility to then remove those nutrients only if you're required to. For instance, we don't have those kind of nitrogen regulations in the state of Michigan. We have some phosphorus. You go to the Chesapeake Bay, it's much more stringent. It varies depending upon where you're living. So we put a lot of energy in, we dilute the heck out of the nutrients by flushing them through with water, and then we put it back in the environment in a reactive form. It's not sustainable. So we want to circularize this. We want to capture some fraction of those nutrients and put them back into productive use, potentially as a food fertilizer. You could also use them as a flower fertilizer. So our work has largely been looking at where those nutrients mostly come from in wastewater. They come from our urine. I'm a wastewater person. We talk about urine. We talk about all kinds of bodily emissions, and that's one of them. So there's about 75 or 80% of the nitrogen is in our urine, 50 to 60% of the phosphorus. More than half of the pharmaceuticals we take end up in the urine. And again, once you dilute them and send them to a plant, they're much harder to recover. So why don't we pull them off, recover them at that point, and allow the treatment plant to focus on carbon and carbon capture and energy production. So that's the vision. There's a lot of technology associated with this, and at the University of Michigan, we are, I'm going to say it, number one. We have the first university-based source separating toilet on North Campus. We have a processing facility downstairs where we convert the urine that we capture into a liquid fertilizer of nitrogen and phosphorus, and then we can separate out, and potassium, those components using different technologies. And then we have done several farm trials in Vermont with our partners at the Rich Earth Institute in Vermont, tracing the nutrients, tracing the pharmaceuticals, and showing that we can produce a safe fertilizer, safer than many of the fertilizers that are used to fertilize the foods we eat every day. But there's also the communication side of this, as was noted by the previous speaker. We have to overcome the negative reactions that all of you are feeling right now about whether or not you would eat that food that was fertilized with a urine-derived fertilizer. And we've done research on the science of, there's a science of disgust, and how we communicate this information to move people towards understanding the benefits and why it's safe. We've also done outreach to help with that communication. And if you were in the peony gardens this past spring, you might have seen a pea on the peonies sign. That was us. We actually have 4 beds. We fertilize 2 of them with urine-derived fertilizer and 2 we don't. Long-term partnership with the gardens to look at the impact, but more than anything, to be able to have conversations with people around beautiful flowers. My second vignette takes us to Flint and my partnership with the individuals in government there, as well as many, many community members who I continue to work with. Even today, some work we were doing on the Flint Community Schools to put hydration stations in the schools, we're ribbon cutting today, which is wonderful. So let's talk a little bit about going back to our water infrastructure and how we finance this massive infrastructure and how water systems are different from other infrastructure we think about. So this graphic starts, this is from the CBO, and it starts in 1977, goes out to 2014, and it shows you the percentage of federal dollars used to invest in the infrastructure itself and building the infrastructure that is water. And the red line is water, and the green and blue lines there are different forms of transportation and highways. So water is considered a local responsibility generally, but in the 70s, after the Safe Drinking Water Act and the Clean Water Act were enacted, there was a massive construction grants investment in infrastructure, a one-time massive investment. And it turns out that we design these, they don't last forever. So there's a design life associated with that infrastructure. And so a tank in one of those treatment plants might have a 30 to 35-year design life. A large pipe that's bringing water from Lake Huron over into a community, big massive pipe, might have a 50 to 70-year lifetime. I'm showing you that we had another massive investment after World War II, and those design lives have come, and we're past them, where we had the largest investments in that infrastructure. That's why it's really important for us to invest in this. So you can juxtapose that then with the cost of water. And if you're in the dark blue line is water and sewer utilities, same timeline. So if you are in a community like Ann Arbor that is high tax base and has large economic benefits, to it, and we have a very nice water treatment system, wastewater system, you can adjust and handle this generally. But if you're in a community that has lost population because industry is lost and your tax base is collapsed, you're not going to put money into that infrastructure. You're going to have a lot of challenges with that infrastructure, and in fact, it's going to be in worse shape than if you had maintained it as you went along, which introduces the challenge with a city like Flint, or like Detroit, or like Benton Harbor, or like Midland, and I can go on. This is Chevy Commons, where they used to build the Chevy. This is after it was gone. You can imagine the loss of that, loss of jobs, loss of population, means a loss of water demand on a system that's already under the ground, already under the street, and was designed back in the 60s and constructed, 60s and 70s. So you have water that's spending a lot of time in pipes because you don't have people using it. That's called high water age, and that's a bad thing. It's a really bad thing from a water quality standpoint. So this is a map of the city of Flint. And what I want to show you is the green and the light green colors are short water ages, 2 to 3 days from treatment plant to your tap. The yellow and pink and orange areas down here are very, very high water ages, up to 3 weeks. You don't have disinfectant residual after that time, after a few days. So this is very poor water quality, and this is independent of lead crisis. So this was already in place. And this map was created by my collaborators at Wayne State, Sean McElmurray and colleagues. So then if you look at where they actually monitor the chlorine residual in the city, those are the red dots, you can see where the dots are not. They're not in the places where you have the highest water age, which also associate with some of the poorest neighborhoods in Flint. You also see what happened during the crisis to chlorine residual, which is not desirable. And so what do we do about things like this? So an example from a research standpoint is we bring in technologies that are accessible to both the consumer and the utility, that provide information for both to improve the situation. A 50 cent or less test strip with a cell phone where you take a picture, goes to the cloud, uses a machine learning algorithm, comes back and tells you it's green, yellow, or red. It's okay, it's not, we really need to do something. And the consumer gets it at the same time as the utility. And now you have a system with granularity in water quality that you can actually take action on. But I think some of the most important work we did when we were there was, and I will quote my friend, Dr. Yana Lambrinidou, who's an anthropologist who's taught me a lot about kind of community-based partnerships, is listen to the community, share our knowledge, and accompany them in the journey when invited to do so. And we were invited to help communicate some water quality related questions they had. They worked together with us to develop a script. We developed a curriculum. It is on the web, and any community dealing with a lead crisis and having to use temporary interim solutions can now utilize that tool that was developed by the Flint community with us in partnership. I know I'm running short on time, so I'll just talk a little bit about Ethiopia work. This is Ethiopia. It's in Eastern Africa. Addis Ababa is the major city, and there are many people involved in this, so I just wanted to make sure I put their names down. Addis is a growing city. It's a city that every time I would fly in, there'd be new oceans of condominiums, big sky-rise apartments, some of which the water pressure was not enough to even get the water to the top floor, and there are no service pumps to get them to the top floor. So people are still hauling water in jerry cans in these buildings. The challenge with cities like Addis, if you look at where their water sources are from, because there's 3 of them here plus some small city wells, the bottom line is that their total supply is about half what they need for just 20 liters per person per day, which is really not adequate for public health. So water does not flow 24/7. What this map shows you is when water is delivered, and there are many places where we work where water might show up once or twice a week. And so the water is stored, again, just like in Flint. In Flint, it's in the pipes. Here, it's in jerry cans and in storage towers on roofs. So you have similar problems and challenges with water quality, and through collaborations with our Ethiopian colleagues and people in public health here at the University of Michigan, we've been able to associate pathogens in the water with diarrheal incidents in children and system issues in the water system and inadequacy of NGOs encouraging people to use chlorine that does not kill the pathogen that's causing the diarrhea. So knowing the system allows us to come to an accurate solution for the community. So I will close by, I want to thank a few people. So my family, so my children are here. They had left school. I know one of them looks like he might have gone, so he's down there. And my husband, who've always supported everything that I've done. And my sister and brothers and my mother, who's no longer with us, but my mother was always my greatest cheerleader in all the crazy things that I wanted to do. And I'm going to close with, these are my graduate students. That is a picture of all of them, except for a few who I don't have pictures of, who do the work, who are wonderful collaborators, just inspiring, passionate, and they really want to learn how to do this work, both of the molecular work and how to communicate it so that they have an impact on the world. And so I feel really fortunate to be here at the University of Michigan to be able to explore that with them. Thank you.
Thanks for a wonderful lecture. We have time for questions. Yes. There's a microphone that will come around. Yep.
Thanks for the great talk, Nancy. In your first set, looking at the urine recovery system, there's a great deal of energy that's expended in producing synthetic nitrogen fertilizer and in mining phosphorus and transporting it from distant places. So it seems to me there's another potential selling point for what you're looking at, which is carbon credits for people who are recycling, developing these closed-loop systems and saving the intervention of electricity and energy that we're now using kind of mindlessly
Yeah.
to.
We've done some life cycle analyses, and we need to do more. But there's an energy demand kind of element of that. And when we compared typical systems in Virginia, we used Vermont, and we used Michigan, urine separation by 3 different technologies won every single time on the energy by offsetting that energy investment that has to go into the complex systems that we have. Yeah, there's more work to be done there, but it's a very good argument. It's that disgust factor and the communication that gets in the way sometimes of being able to convince people of that.
Great, we have a question online.
I have a question from our YouTube audience. They ask, can you shed some light on why our regulatory requirements for supply and quality were built the way that they are, that they have now led to these systematic problems and inequities?
So the regulatory requirements and how those have led to what we have today in the challenges. Well, you know, actually, I think many of the regulations were very practical, certainly at the time that they were developed. There are some regulations that are completely inadequate. The lead and copper rule is a great example of that, and it's well known. And now it's a political turmoil in terms of changing it and fixing it. And so I think sometimes what happens is if you get the wrong elements in the room when those regulations are developed, you can have that. But many of these regulations are actually quite stringent. The Chesapeake Bay nitrogen and phosphorus regulations are among the most stringent in the world. They've actually improved. They finally took a while, but they've started to improve the quality of the bay. So you see shellfish coming back, and so it can make a difference. It absolutely does make a difference for the environment and public health. I think it's when those regulations are not, or a system isn't adequate enough to be able to comply with them. And it's the kind of investment disparity is what I'll call it in the way in which we invest in these systems that's failing us right now. And we have to bring some equity into how we upgrade systems in those cases. So it's a complex question. I appreciate it. And I'm sure whoever it is can contact me. We can discuss it more.
I have a question which is related to the last part of the talk. I had the sense when you're speaking about water systems in Ethiopia that some of the received ways of doing things in the field are not the right approach given what that water system looks like. Could you expand upon that a little bit? You actually even mentioned something about chlorine that I didn't quite catch that would be interested in.
I was moving through that. So you have a centralized system in a country that does not have the resources to maintain it. That's the bottom line problem. And so one of the things that motivated me to develop a course on decentralization here at U of M was looking at the textbooks on the professor shelves in Ethiopia, and they were all the same textbooks as I have on my shelf. And they're the wrong systems for what their country needs. So part of the problem is we're not looking at where those countries are at this time and their ability to actually put the resources into them to maintain them. Some countries have moved from low to middle to middle income and been able to do that through centralization. South Korea is an example of a country that really went through this process and has very high sophisticated systems today and people living in density and very healthy. But that's not the case in Africa, and it's not going to be for some time. So we really have to get back down to basics and identify what those solutions will be. So I don't know if that gets entirely to your question, Mike, but that's the starting point. When we went up to the utility, they had a treatment system just like we have maybe in Ann Arbor or in Detroit. But there were cows at the intake, which are putting in cryptosporidium, which is the source of the diarrhea for the children. They're not optimizing their coagulant, their filters are short-circuiting, and they're using chlorine, which doesn't kill crypto. And then the NGOs are adding chlorine tablets at the homes with people in the jerry cans when they don't understand that crypto is a key source associating with childhood diarrhea. And they don't, so you have to have the system understanding to be able to put all those pieces together. And that was really great to do with our Ethiopian partners because we had people from public health and nutrition and engineering working together who hadn't met before.
And we're now working on their system in their city to understand that.
Thanks. That's really helpful. Thanks to understand. Well, at this, let's thank our speaker, our distinguished university professor, for a wonderful lecture. And now I'd like to just take a brief moment to close out our symposium. This has been a wonderful event. I've really appreciated both the virtual and in-person participation, and very grateful to our three distinguished faculty member speakers, and would like to congratulate you once again. Please join me. Thank you. Have a good afternoon. Thank you.